To the OHBM World Health Organization Symposium. The purpose of this symposium is to raise awareness among the participants of OHBM, especially that of young researchers, to the need of current research projects that impact upon the staggering burden of disease, of brain disorders. We also hope to stimulate discussion around this topic. It's a pleasure for me. Next. It's not functioning. Can I have the next slide, please? Yeah, just a moment. It's a pleasure for me to chair this section together with Dr. Shekhar Saxena. He is director of the Department of Mental Health and Substance Abuse of the World Health Organization in Geneva, Switzerland. And it's an opportunity to say many, many thanks to the organizers, to Pedro Valdez Sosa from the Cuban Neuroscience Center in Havana, Cuba, to Christophe Michel, Department of Neuroscience, University of Geneva, Switzerland, and to Sean Hill, Swiss Federal Institute of Technology, Lausanne, in Switzerland. And now I would like to give the word to Dr. Saxena.
Thank you very much, Carl. And to begin with, I would like to thank OHBM to have given us this opportunity to address the issue that is top of at least our mind. And by the end of this session, I hope in the minds of at least some of you. The issue is, can and in what way brain and neuroscience research contribute to decreasing the public health impact of mental, neurological, and substance use disorders in a short time, and especially in those situations where resources are extremely limited. This is the question that we're asking today from a number of very high-profile researchers and investigators of brain projects which are working in a very dedicated manner for a number of years in this area. I must say that personally I feel a little bit intimidated in the presence of many of you who have spent your life solving, or at least trying to solve, the mysteries of the brain, which I don't claim to understand even at a very basic level. I'm a medical scientist, I'm a psychiatrist. I did do my neuroanatomy and neurophysiology, but that was 30 years back. And I'm sure things have changed a lot. So I'm here to learn. I'm here to learn from you about the kind of research that is taking place. But I'm also here to provoke some questions as to how that research relates in a shorter term and with a clear angle to how it will benefit the millions of people who are suffering from disorders of the brain, in which I include the disorders of mind, the mental disorders, the behavioral disorders, the actual brain disorders, and substance use and other developmental disorders which originate in some way in the brain. I hope to go back from here in a few hours, learning a few things, but also finding a little bit of a way to how this collaboration between brain scientists and public health people could be strengthened, and finding in few directions about how it could be done. Again, we are very grateful to OHBM to have given us this time, and for all of you to be here, to work with you, to try to answer some of these questions. This is the first session of 2 that we have organized. The session is the first one. We're organizing a closed session tomorrow. This is an open session, but we are having a closed session tomorrow within WHO, in which at least some of you will join. And we hope that this session will put the background against which we can make more progress tomorrow, and then find a way to work together in the coming years. So with that, I stop here and hand over back to my co-chair to begin the presentations. Thank you, sir.
Thank you very much. I have to announce the speakers of this symposium. And the first speaker is Walter Koreschetz for the Brain Initiative, Director of NINDS, Office of the Directors, Bethesda, Maryland.
Thanks. Thanks very much. I have 10 minutes to tell you about the Brain Initiative, and I'll try and do that as fast as I can, but concentrating on what I think is the implications for health in neuromental and substance abuse disorders. So the caveat is that at the National Institutes of Health, the largest amount of money of any area goes to neuroscience. So neuroscience gets about $5.4 to $5.5 billion of research funds every year. The Brain Initiative is a component. It's about $150 million a year. It's substantial, but I'm going to concentrate on purely the Brain Initiative today, but clearly there's so many other things that are going on funded by the taxpayers in the U.S. that are going to help, hopefully help neuromental and substance abuse disorders. So start out, this is an article by Pam Collins in NIMH looking at the global burden of mental, neurological, and substance abuse disorders. And these are the largest cause of disability-adjusted life years in the developed countries and are becoming as equally important in the underdeveloped countries. I have little arrows next to the disorders that have known pathology. So when you look in the brain, you can actually see a pathology underlying the disorder. But as you can see, most of them, you look in the brain now and you don't see anything that is an indicator of what the problem is. And we'll get to how that intersects with the Brain Initiative. So the Brain Initiative was presented by President Obama in the State of the Union address in 2013. And this is a major initiative that he asked the federal agencies to engage in, and the BRAIN stands for Brain Research Through Advancing Innovative Neurotechnologies. And the idea behind this is that scientists, and this is not news to anybody in this room, is that we're all interested in trying to get a dynamic picture of the brain in action, but our tools really don't allow us to really sink in and see things as they're happening. We have tools, but they are feeble in terms of what we know is going on, the complexity of what's going on in the brain. This is an effort that NIH is leading, but also there are multiple other agencies in the United States government, particularly the National Science Foundation, which we work closely with, and there are other private organizations that are also working with us on this Brain Initiative. So the Brain Initiative, if you're interested, it's outlined in a report called the Brain 2025 report, which was a quite scholarly document indicating how the U.S. could develop the kind of technologies that would kind of revolutionize how we are able to investigate brain circuit activity. So the key here is the focus is on circuits and networks. The key is on trying to get better tools to measure the fluctuating electrical and chemical patterns within the circuits, and then to use this information to understand how all this helps generate our unique thoughts and actions. So the translation to health, I think for people who have thought about this, is really quite easy. Let me try to give you my opinion. I think for the disorders that I mentioned earlier, which have no pathology when you look inside the brain, they are clearly primarily circuit dysfunction disorders. And the people in this room are really the pioneers who try to understand those circuits. And so understanding and be able to measure, to be able to manipulate these circuits is really at the core of getting at not only the diagnosis, but also the treatment of these disorders, and also potentially to get us to understand what these circuit problems are that we can perhaps actually prevent them from causing disability if we can capture them earlier on. But even for those disorders that I deal with in the NINDS, where there is a pathology in the brain, the patient is not really disturbed by the fact that they have alpha-synuclein or amyloid depositing. What their trouble is that their memory circuits aren't working, or the motor circuits or the autonomic circuits aren't working. So even there, our focus has been on the molecular and structural pathology, and then we have jumped this black box, which is the circuit abnormalities, and tried to give treatments to patients, wait a couple of years and see what happens without any ability to detect what that intervention is having on those circuits. But just imagine if we had information, the ability to interrogate those circuits, we could actually, I think, much more quickly come to beneficial medications for the circuit disorders that are causing the patient's problems. I think the talk this morning on schizophrenia basically outlined just how complex these disorders are in terms of the circuit dysfunction, and how our therapeutics completely ignore that kind of complexity. And perhaps that's why they're so poorly effective, or why they come with such side effects. So the problems that we're trying to face in trying to get treatments for patients, these are really tough problems. So we are not in an easy situation. It's not a matter of trying to understand how to kill a bacteria. We're trying to understand how to get people's brain circuits back to working again, or preventing their deterioration. And without those tools, we're really stymied. And so how does progress come? Well, I'm very taken by this remark from Freeman Dyson, that if you think about science, the new directions are launched usually by new tools more so than by new concepts. And I think that's also true in medicine. So when I came out of medical school in '74, we were very excited to see this CT scan, the first time we could actually see the brain in living human beings. But I'm sure the people in this room who are younger than us look at this and laugh. Look at the kind of detail you can see now in 2012 with high field MRI scanning. And this certainly revolutionized how we practice neurology, the ability to actually see structure in the brain. But our functional ability to look at function in the brain is really not much different. If you think about the major tool that's used in neurology, it's the EEG, which is a 1928 technology to get at brain activity. So what is the Brain Initiative? How do you break it down? Well, we have kind of a roadmap, and we have broken it down. So the first tenet is to try and understand the diversity in the brain. And this is the parts of the circuits. What are these parts? They're the different cells, these different cell types. We know neurons and glia, but as you know, and certainly have seen in talks today, there's not one type of dopamine neuron. There's lots of different types of dopamine neurons, and we don't have a way to identify them very easily. We don't actually even be able to go in with imaging and tell whether it is decreased neurons or increased glia. We see a water signal, and we infer that it's gliosis. But why don't we have better imaging technologies to actually get us at the cell types? This is the crux of the Brain Initiative, and that's to map the brain circuits at multiple scales. So people here at these conferences, you're looking, particularly at the imaging, you're looking at a meso or macro scale. But as you know, you can go down to the micro scale. So there are people working on serial section electron microscopy of neural circuits. Incredibly data dense, but it's potentially going to be doable in the fly brain, in the zebrafish brain. And there we're going to have an unbelievable microstructural detail of the circuits, the wiring diagram, and then try and put function on top of that, I think, is the next step. So the brain in action, trying to get tools that will allow us to see the functional activity. Bold technology, which certainly is a mainstay of much of the discussion here at this meeting, is a surrogate for the brain activity. Wouldn't it be great if we could get closer to that, or really understand what are the neurodynamics that are underlying the bold? That's actually one of the projects in the Brain Initiative, to try and better understand that. But finally, to go past the ability to just correlate behavior with some imaging signature, to get beyond correlation, but to actually then intervene and be able to really know that the circuit you're thinking of is really causally regulated as you think it is with behavior. You need to intervene. You need a model that when you do intervene, you make the right prediction in terms of how the behavior will change. So demonstrating causality. Now, the other part to kind of point out is that most people feel that our understanding of how the brain works is based on such limited knowledge and understanding of the activity patterns that we have no idea of what the rules are that are generating network activity and how network activity is generating behavior. Because much of our knowledge is from single cell recordings, and people have said that's trying to understand a movie by you get to see one pixel at a time throughout the whole movie, and then you can run it again and see another pixel. How long would it take you before you could figure out what that movie was all about? Probably never. If we have the ability to actually record from thousands or millions of neurons continuously, that's what's going on in the brain. And certainly the imaging data shows you the brain is lighting up in a very distributed fashion. And if we could get that kind of data, the first question is, how would we analyze it? What would we do with it? But the main point to get to is, what are the rules that are in that kind of network activity that determine behavior? And that's, we think, are completely new fundamental principles that are going to come out of this. The purpose is to advance human neuroscience, to understand the brain and treat its disorders. And I think we can make a strong case that some of the technologies in the Brain Initiative may lead to therapies or better understanding 20 years from now, but we are also investing in things that are cutting edge that can be brought to patients in a short time frame, like non-invasive stimulation of the brain or invasive stimulation of the brain that occurs in deep brain stimulation, or recording from the brain that occurs now in people with epilepsy, and trying to understand where their seizures are coming from. To learn more, I'd refer you to the Brain website, where we have all of grants that are funded and descriptions of them. And I just wanted to throw out a couple of the advances that are coming out of the Brain Initiative already. So just some examples from Dr. Stanley at Mount Sinai, the ability to label trip channels in the hypothalamus, specific neuron groups in the hypothalamus, and activate them or deactivate them by radio frequency waves from outside the head. And this is cells that regulate metabolism in the body. Focused ultrasound, an amazingly interesting technology where you can hit a 1 millimeter spot anywhere in the brain. You can create a lesion, you can open up the blood-brain barrier, and maybe you can safely stimulate or inhibit that circuit. Think of what it would be like if we could try to understand the circuitry in the patient that's depressed with a safe mode of targeting particular areas inside the brain to find out if turning on or turning off them causing dramatic symptomatic changes. I don't have time, I'm running out of time, but the DREAD technology, getting genes into particular precisely targeted neuronal regions that then are activated by a drug. Interesting idea that could be the future. People, certainly in Parkinson's disease, are thinking of doing this to activate dopamine neurons. The Connectome project, I won't mention, but to say that tremendous amount of work and data has gone into this, I think certainly helps a lot of the people in this field. There are people working in neurorehabilitation, trying to understand how the brain rewires after stroke. And then, there is this amazing, not population health certainly, but people have brain-computer interfaces that now allow them movement that they never had before through activation of robotics through their brain waves. So this lady was paralyzed, and if you could just imagine what it's like to be paralyzed for years and then all of a sudden have some ability, any ability. It's really her smile tells it all. And then the last thing I'll say is that the Brain Initiative, I think, will tell us things that there's no way we can predict right now what they're going to tell us. And that's the beauty of science, particularly at this level, that the things that fall out have dramatic impact in places you could never know. And this is just one example where one of the Brain Initiative investigators who's trying to do the cell census in the frontal cortex in fetal brain found a receptor on the radioglial cells, which he believes is where Zika is entering the radioglia and causing the microcephaly in the babies born to people who have been affected with Zika. So again, an example, who would have guessed that the Brain Initiative was going to have relevance for Zika, but here it is. So thank you very much for your attention. The points I'd like to leave you with is that The ability to monitor brain circuit activity will lead to objective biological diagnoses to inform more precise use of treatments. That's going to be good for the public health. The targeting the normalization or compensatory circuits will lead to more effective pharmacological treatments, less side effects, new treatments via invasive or non-invasive direct modulation. That will have major public health impact. And the new knowledge will lead to advances that are now completely unpredictable. So thanks very much.
Yeah. Okay, if you have a few questions, we have time for it. Pedro.
This is rather a comment and a question, and it's valid for all the rest of the brain projects. I see all these exciting advances, which I think are going to be fundamental and change. This is the opening into actually solving many of the problems. But I'm concerned that sometimes these projects, including the brain project, neglects translational aspects. So, for example, you were referring, Walter, to the old technology of the EEG. Now, it's very simple. The Connectome project records everything except the EEG. And there's evidence, we've published it and other people have worked on this, that you could use this as a translational tool and calibrate, not as the definitive diagnostic thing, but you could calibrate these, let's say, lower-end methods. But something that's missing, I think, in all the brain projects, simply because sometimes we don't think about it, is how can we make some of these things available, even as a first level, let's say, and then it fits in with the rest. And I think this is, why am I pointing out? Because I think precisely this is something we can discuss about in these 2 days, because it could be done at very little expense.
Yeah. Yes, I agree. And as I first mentioned, that the Brain Initiative is only a piece of what we do at NIH. And I would say in comment that, there's a lot that could be done if we had a gold standard, and then we could look at different technologies to see if they give, how much information is there lost versus using a lower level technology versus the gold standard. For certain problems, you may not have any information loss. An EEG may be as good as the gold standard. But on other cases, it may be not as good. And without knowing that, you can't actually use the technologies that are available to you. So that's kind of the generic way I'm thinking about this. And I think that there's also room to move the more intensive technologies to try and understand how to extract the information using much more inexpensive and accessible technologies. So I think that's another area where we can talk more about. And finally, I'd say, in terms of the EEG, we actually are very interested in trying to get people to study what is the neural activity that's generating the EEG components, and to actually get better information to inform how you interpret EEG data. So actually, that is a Brain Initiative project. Thanks.
Thank you. More questions? Presently not. Then we come to... Oh, yeah.
Very interesting, thanks. I was just wondering kind of like what is the financial breakdown between the different parts where you're going into this thing? Because I was wondering, the director, forgot the name, mentioned that he didn't just want interventions, he wanted cheap interventions quickly. And I was wondering, where are we spending the most money? Are we spending appropriate amounts of money also on things that can be done cheaply, that may not even need pharmacological intervention, or may not need expensive tools? Are we still looking at behavioral intervention, which could be done a lot cheaper in in places other than the developed world when we don't have to put in a $1 million MRI?
Yes, so I'd say that behavioral interventions, the role of cognitive behavioral therapy in multiple different areas is an entire area of intense research at NIH. That's correct. It would not be something that would be in the Brain Initiative, which is based on developing neurotechnologies, but it would be a very kind of active area of research.
Sorry, in that case, could I follow up by asking, so why is that not combined? Is the idea that what the environmental impact on the brain and the behavioral impact on the brain is not big enough to be worth that?
No, I would say that the Brain Initiative the idea is to develop the technologies that anyone can use. So I know we might have 10,000 investigators that we support in this space. The Brain Initiative investigators might number 500. So they're supposed to build these technologies, and then we will actually disseminate them to all the other massive, much larger groups that are studying the kind of problems that you talked about. So the idea is to raise all boats by building these technologies to allow people to do better science, to get answers that are more precise, and there's no, I mean, the issues that you've been mentioning are, they're brain circuit issues that we don't have the ability to measure the circuit activity. So I think it's going to help a lot.
Just to answer, like Michael Garwood's group is building a portable MRI that's liquid nitrogen cooled, so you don't need the expensive helium. So there are projects that are addressing that.
I think all of the people who have spoken are thinking of the interface between the Brain Initiative and other work.
Yeah.
I just want to add, and I think that you've pointed out correctly, no? The thing is that I want to add another component. Where most of the people that have brain disorders live is in poor countries and in low, middle, and income countries. That's where least research is going on. So if I may suggest to all the projects, and I'm sorry, just that you were the first, we should think of doing translational research in these areas so that the benefits of the brain projects, so that in some way be adapted to the conditions of countries which have totally different situation from that where the major areas of research are.
No, I agree, and absolutely, we should be able to talk about that hopefully tomorrow. And I'd say, NIH does invest in brain research, neuro research, psychiatric research in the underdeveloped world, and you have to be realistic about what the needs are. So you can't, the Connectome MRI is not gonna, you're not gonna see one in underdeveloped countries for a long time. But the information that comes out of that should be able to be translated into getting better health everywhere across the country, across the world.
I think there are two different perspectives on this problem. One perspective is the research problem, and there we should use the best and most advanced technology we have to get a definition which fields are promising to further develop by downscaled technology, which is everywhere available, also in low-income countries. But these are two steps, and here we speak of the first step to clearly identify aspects which must be further studied, eventually with additional research support to downscale high-tech instruments to instruments which can be used in low-income countries. One short question, then we have to go on.
Okay, thank you very much for your talk. It's just a quick question. Like, what is being done in your institution about promoting these ideas to people that they need to do translation and stuff? Because if you see decision making had the room full of people, and today we have something about how to apply this into collaboration between the WHO and the OHBM, and like there's just a few of us. Thank you.
Wait, maybe you better redefine it. So you're asking, what do we do to try to move science to translation and then to patients? That's the question?
No, like what are you doing in order like to make people aware, like in your institution, that there's need of help elsewhere, like how that they need to think about translation and that stuff, because it seems like most of us are not interested in that.
Well, I'm not sure about it. I do know a couple of people who I've seen here who are interested in translation. But I think, yeah, as I mentioned before, the neuroscience effort at NIH it's larger than anything else. And so it's got really strong pockets in every single area, from, work in the undeveloped world to high-end imaging to trying to develop therapies that go into patients. So at our institute, we have a virtual pharmaceutical company type of situation to help investigators bring products to either diagnostic IDEs or INDs from the FDA to go into patients. We have networks set up to test things in patients. So if you're interested, we could kind of go into what else NIH does with the $5.5 billion of neuroscience money. Great.
Thank you very much, Walter.
Yeah.
I'm sure there are many more questions and comments, but I would request you to hold on to them, listen to some of the other brain projects, and we can again come back to several of these questions to work out in more detail. It's my pleasure now to invite Mu Ming Pu from the Chinese Brain Database to make the presentation. Are you here?
Mu Ming Pu? It's a video. Okay. Can we get the video? Yeah, moving. Yeah.
It seems the video is not ready, so let's move on. Catherine Aumund's European Human Brain Project.
Hello, everybody. It's my pleasure to introduce a Human Brain Project to you, and I hope I can show you during this talk some applications that are interesting and important with respect to health and to developing tools for diagnosis and treatments of neuropsychiatric disorders. And before I would like to start, I would like to come back to the notion that the brain has a multi-level type of organization. So it's organized on different scales, both in space and in time. And you see here on this image some of the different aspects that contribute to human brain organization. For instance, the distribution of receptors for neurotransmitter systems, cellular architecture, fiber architecture, and other aspects. And I mean, the scales which these aspects cover, they are really quite different. So we start from the nanometer scale when we think about molecules, and we end up with several centimeters when we consider large and long-range connections within the brain. And all these different aspects, they contribute to brain organization, and they make it so difficult and so complex to understand how these different aspects work together and how they are, for instance, disorganized when we think of human brain diseases. So the HBP is a European flagship project which was organized by the unit called Future and Emerging Technology of the European Commission. And the major aim of the European flagship project is to contribute to an understanding of the human brain, which is, in our view, one of the major challenges of the 21st century. And if we can have this profound understanding, then we, of course, will better understand what makes us human. We will contribute to basic science, but we can also contribute on a middle and long run, at least, to develop new treatments for brain disorders. And we are convinced that we are now in a position where the ICT possibilities are mature enough in order to reach this goal. So one of our major objective is to put in place a cutting-edge ICT-based scientific research infrastructure that will permit scientific and industrial researchers to advance our knowledge of the fields of neuroscience, computing, and brain-related medicine. So it's not so much a neuroscience project. It is really a project about creating this infrastructure, which can then be used by neuroscientists, but also by other scientists, and also by engineers, by computer scientists, to promote our knowledge. And the project contains 12 sub-projects which have a different focus. So the Human Brain Project is one of two existing flagship projects of the EU. It has a 10-year perspective, which is really a long time and which enables to ask questions on a longer run, and this is this is very important. All of you probably have these 2 or 3 years research projects, which are, of course, very limited, both with respect to funding, but also with respect to the time scale. We have about 400 researchers from 114 institutions, 24 countries, and you can imagine that it is quite hard to set up this project in such a way that all these people collaborate with each other and also produce an outcome that can be then delivered to the science community. So presently we are negotiating with the European Commission the first phase of the operational phase. This is the next 2 years. And we expect to have a budget of approximately 89 million for these 2 years for this part of the project. So how the project is organized? We have, as I said, 12 sub-projects. One is central services. We have 4 neuroscience oriented research projects addressing the mouse brain organization, human brain organization, systems and cognitive neuroscience, and theoretical neuroscience. Then we have 6 so-called platforms or infrastructure sub-projects addressing the neuroinformatics platform, brain simulation, high-performance analytics and computing, medical informatics, neuromorphic computing, neurobotics. And we have an ethics and society sub-project addressing responsible research, consequences of such research for the society, ethical questions, practical but also theoretical ethical questions. And I will focus very much on human brain applications and their implications and a little bit on medical informatics. Let me just shortly comment that brain simulation is one of our key aspects, which is perhaps unique in a certain sense. So here we try really to develop simulation tools on different levels, starting with molecular simulations, going to cellular simulation, and also to larger scale simulation. And in many other fields of research and science, like astrophysics, for instance, hadron physics, and so on, there are very powerful methods of simulation, and perhaps this is one way to advance our knowledge about the difficult and very complex interactions in the brain, for instance, on the molecular level or on the cellular level. We have neuromorphic computing platform, which opens up quite interesting possibilities for simulation, because these neuromorphic devices, they are some kind of analog computers, and they can speed up simulations in a very sophisticated and very crucial way. So, for instance, when you would like to simulate plasticity and learning processes existing technologies, classical technologies, are far too slow in order to capture this time scale, and they can do it almost in real time, which is, of course, an exciting possibility for all those interested in learning and plasticity. And we have a neurorobotics platform, again, which is very much simulation-based, but I think it is very important to have robotics because at the end, the brain is not just an organ. The brain has a periphery, yeah? So it has a body. And to simulate also this interaction between the brain and its periphery is a necessary prerequisite in order to understand how the brain is working. So let me give you just a few examples, and one comes from the receptor architecture, and the author of this research is sitting next to me. So Carl Zillis, his team, and me, we are trying to better understand the receptor architecture of different receptor binding sites in the human brain. And the special thing is that we not only see for a single receptor type or transmitter system, but for a whole spectrum, because we are convinced that it's a balance of different receptor types that is important and interesting, and that is in disbalance when we think about epilepsy, for instance, or other neuropsychiatric disorders. And here we produced quantitative data, which are then delivered to an atlas and provided to the science community, which quantify the distribution of receptor types in the human brain. We start with certain cortical areas and subcortical nuclei, and these data can then be transferred, for instance, to patients here with epilepsy. Another really crucial problem, and this is connecting to what Walter Korschitz was saying, is of course the connectivity. And in the human brain, we are in a very difficult situation because mapping the human brain and understanding how the connectivity is very much based on studies on experimental animals, on tracing studies, for instance, on studies of ontogeny or on diffusion studies, but these studies are, although performed in the living human brain, they are limited with respect to the spatial resolution. So we have here developed an approach based on polarized light imaging where we can go into very much detail to a scale of 1.3 micrometer, identify single axons, and at the end we hope to come up with connectivity maps, structural maps of the human brain, which cover the scale from the micrometer to the centimeter scale. And what you see here in the background, these are really single fibers, and when you compare it with the present slide, this is approximately the resolution that you have in vivo. So linking these different scales is one of the major aspects that we would like to address. So having such data means that we should bring the data into a common atlas system, and there are different atlas systems available in the world, and I think to have a common understanding how this atlas should work and what are the linking points to other brain initiatives, to other sources, this is an important step in order to bring these data together. So it's not enough to have a good database, one has to have a good atlas which reflects the different modalities of data, and therefore we should identify links, of course, between the different data formats and also links between the different modules. And in the HBP, we are developing an atlas which is available through the neuroinformatics platform, through the collaboratory. There are data also available, not yet now, very easily, but in the future, coming from patients. And there are extraordinary data coming, for instance, and showing intracranial EEG data from different patients where we record brain activity and compare it, for instance, with functional MRI signals in order to bridge really this gap and to better understand what is the nature of the EEG signal and what does it tell us, for instance, about brain activity. So this is an example showing just how we can combine different aspects of brain organization in order to address questions for patients. So we analyze, for instance, the cytoarchitecture of the human brain, that is a cellular architecture in different brain regions, and FP1 and FP2, these are two areas which we have identified in the frontal lobe. And the frontal lobe is, of course, very important for our social behavior. It's a very newly developed cortical area in the brain during evolution. So when we map this area not only in one brain but in 10 brains, and this is necessary because the extent of the areas and also brain shapes are very different between subjects, and intersubject variability is one of the biggest challenges that we have when we address morphology and function of human brains. So when we map these in 10 brains, then we can create probabilistic maps and use these maps as a mask to better understand the connectivity of the brain, which we can analyze, for instance, in patient or in healthy controls. We can see how the brain structure has been changed, for instance, in patient with major depression, as we did it here, and the superimposition of the cytomaps with the data coming from depressive patients shows that there's an area in the midline which is changed as compared to controls. And when we then link it to gene expression data coming from the Allen Brain Institute, then we see that indeed there are genes involved which are relevant for certain transporters, for neurotransmitters. And this somehow closes the loop so we can come up with better ideas about the pathogenesis of brain diseases. To have a more practical application, there's a medical informatics platform which is also available through the collaboratory. And this is at this moment only a first and very rough and very preliminary version, but still the idea is that in this platform we make available data coming from patients from different hospitals, and these data can be then analyzed, for instance, using deep learning and machine learning algorithms. And the group here around Ferhat Kerev and Mira Marcos, they have identified two different types here, an atypical Alzheimer's disease types and a more amnestic type based on many features analyzed by deep learning. And the idea is that we can come up with a more biologically based classification of neuropsychiatric diseases. So I think that the brain is much too big and also brain diseases are much too complicated in order to address this question within one initiative. And I share, I think, this idea with many of the colleagues here today. We need to have established the links between the different brain initiatives, between the tools and the different databases, and then should be able also to make this very important translational step to health. Thank you for your attention.
Questions?
Yeah, a few questions.
Although we'll have time in the end for further discussion, if there are very specific questions related to this presentation, just maybe 2 or 3, they can be asked now. I don't see anybody. In that case, thank you very much.
We can go on to the next presentation by Tetsuro Yamamori from the Japan Brain Minds Project. Dr. Yamamori is working at the Riken Brain Institute in Tokyo.
Oh, okay. Not for me.
No.
So what can I do?
No. Can
I can.
Can I ask you to share the place here
sure.
us, and
No
we
problem.
see the video and then your presentation?
Yeah, it's my pleasure. Yeah, no problem.
Yeah, sorry
Please join us here.
for that. Okay, the China video. No.
Yeah,
Please.
Okay, thank you very much. My name is Tetsuya Yamamori, come from Riken BSI, and on behalf of Hideyuki Okano, Keio University, he's not come to here. So I would like to explain our brain-mind project in the mammoth brain in Japan. Can I go next? How can I go next? Oh, yeah, push this. No, this. Okay. Brain mind mapping project, as you heard from the United States and also European brain project, and our is focusing in marmoset. The mammalian has about 5,000 species on the Earth and has a own specific brain structures, as you can see in this website. And particularly in primate lineage has a particular feature, as you can see, which is different, for example, most well-done mammalian model systems such as mouse. And that has a specific feature of anatomically distinguished properties. We are particularly focusing on common marmosets with several reasons. One is its small size and relatively easy to handle, and also high reproduction ability, and also more importantly, we just generate transgenic marmoset as published in Sasaki and Okano's in 2009. And this is published in Nature as an article, and this is first German transmittable transgenic line, and which enable us to manipulate in a primate to some extent close to mouse. Now, so this is the reason we started this Japanese brain mind project a couple of years ago. And this is a name we said mind that stands for brain mapping by integrated neurotechnologies for disease studies. And that we call brain mind project in Japan. And these are the three structures and two as a project. One is structure and functional mapping of marmoset brain, and second is that B, development of innovative neurotechnology for brain mapping. And this is A and B. We made a core institute in Riken, and that is we called core institute. And the C project is human brain mapping and clinical research, which is led by Professor Kasai in Tokyo University, psychiatric professors. And so the project leader of Project A is led by Hideyuki Okano and in Keio and Riken. Also Project B is led by Atsushi Miyawaki in Riken. And also the C, as I mentioned, is led by Professor Kasai. Now, let me explain, first of all, the structure mapping using technologies, different technologies. One is the MRI, and the second is that I'll explain about the tracer injection and also two-photon microscopy. And the third one is the electron microscopy. This is a very different level of analysis, which is coming from this order to 1 millimeter to MRI to up to 100 micron or more or less by two photon and tracer injection, and also finally electron microscopy level is to 1 nanometer. So it's about 10 to the 9th order of difference how you can integrate all those information into the brain map. So that is kind of our challenging, and that's as mentioned from the European brain mapping, the important thing is that we have a huge amount of MRI information in the United States and Europe and other countries. And then how to this one combine to marmoset project is one of the issues that I would like to explain a little bit about. The effort by doing in Riken and Okano's group, and we have set up this 9.4 Tesla MRI in downstairs of Riken, and then we have obtained the images, and this is one of the pictures, and then specifically used for marmoset. Now, this is one of those mapping by Junichi Hata in Okano's group by using this MRI imaging, and then how these areas connected in marmoset. This is actual data driven by MRI data and to set up the two-dimensional maps. And this is further expanded in V1 and visual areas, how these are connected. And also recently, the Okano's group published the myelination in MRI is also can be useful. So this can be sort of matched with histological analysis, which has been mentioned by European brain mapping project. Now this is, I'm not sure I can, I don't think, can I?
Is this a video?
Yeah.
Yeah, can you
move on?
Click
this?
on?
Yeah, thanks. Oh, thank you. Yeah, you can see this. This is, we just get mice of injecting one dye to prefrontal cortex and then running to this cut with two-photon tomography and then reconstructed the brain. So you can see how these marmoset brain looks like. It just injected to area 9 and then how these brain areas are constructed, in particular in marmoset. There's a typical structure which you can see in most of anatomy, but also there's a kind of structure you can see in specific in marmoset. So this is the mapping strategy, but we also do these transgenic marmoset approaches and also knockout marmoset approaches led by Sasaki and Okano's team. And we are now generating these disease models such as Parkinson's and also Alzheimer and also autism model. And that is being made and to analyze to understand what is the molecular and cellular mechanisms that cause these diseases. And also the Tomomi Shimogori in Riken, who is now doing the in situ hybridization using marmoset brain. And this is just one of the examples, but she is now ready several hundred marmoset in situ brain and will be open very soon. We are working to open to public, and then that will be available pretty much soon. Now, this is another approach used by MRI in Kaneko in Okano's lab, and Takaki Kaneko, Okano's lab. And this is sort of the compare with statical MRI and also those functional MRI, and to review what particular brain region is activated in particular visual tasks or other tasks. And that's kind of a comparison. This is the issues how this resting state activities related to actual active state of the brain state in comparison with MRI. And also, let me explain a little bit about the Project B, which was led by Atsushi Miyawaki in Riken, who is one of the leading researchers in fluorescent protein who works with Roger Chen and Roger Chandra. And also, he also invented recently one of the pioneers to use transparent brains and then combine with the fluorescent technique and transparent brain and to apply this possibly marmoset. And then we can further identify more detailed structure in combine with these structure mapping. And finally, to particularly relevant for this symposium, how we then organize this kind of basic approach to clinical approach. So we have set up this Project C led by Professor Kyoto Kasai, and that's the central team we are having the core institute, namely Riken, and led by Hideyuki Okano, and I served as a vice project leader in Riken. And then this is sort of collaborate to clinical people led by Kyoto Kasai. And then that's Project C has three structures. One is Hitoshi Okamura, and that works neurodegenerative programs and also psychiatry programs, and led by, again, this is Kyoto Kasai. And also we have neurovascular diseases to solve by using marmoset model. This is led by Ryosuke Takahashi in Kyoto University. And these three teams combine together and then to work with each aspect to use marmoset as a model system. Our strategy is the starting first one is to identify disorders, symptoms that is particularly find both in human and marmoset. And then if we can find particular disease that is relevant to human diseases in marmoset, we further study using this project stage 2, so develop particular strategy or more the mechanism how these diseases cause. And then eventually we hope to come up this to some say more deep in neurocircuit level, and then that finally to feed back to clinical approaches. So this 1, 2, 3, 4 feedback we hope that we can contribute back to clinical application to solve the fundamental questions. So this is about our Japanese brain mapping project, and you can find this more details, particularly in the Brain Minds homepages that's shown here. And I thank you very much for your paying attention.
Thank you for sharing this information with us. And there is a question, I see.
Yeah. Maybe I missed it, but is the primary data being made available for others? Are you making the data that you collect available? The
Yes.
primary non-manipulated data? Because I think that's where these projects will lead to some of the greatest benefits.
Yes.
Okay, great.
Yeah, that's our intention. And we are hoping to several stages, but first stage, and hopefully next year, up until next year, we can open this our data to the public, entire world. Thank you.
No more question? Or,
Pedro?
yeah, Pedro.
Is there any major component about cognitive aging and dementia? Because I didn't notice it. I saw neurovascular, which of course is an important component, but considering the aging population everywhere, in Japan they have very good health and people live a lot longer. Mm-hmm. Wouldn't they be? Isn't there a component for this?
Yes, it is.
Okay.
I didn't go to detail, but it's of course in part of the clinical teams that's focusing on aging, and we also want to use the aged marmoset to compare them. Thank you.
Okay, and now high-tech meets a high-tech country and a high-income country. We will see after that. We will see the Chinese video. Maybe. Aha. After the last talk, we have a general session for questions.
I would like to present to you a brief introduction of China Brain Project. This project was approved by the Chinese government earlier this year, and it's expected to be announced later this year. And in this brain project, which I was one of the chief organizers, the Chinese scientists and engineers have come to a consensus that the project will consist of 3 components. The main components would be to understand the neural basis of cognition and to develop brain research technology platform to facilitate these basic studies. And importantly, there are 2 arms for this project. One is to develop an effective approach in early diagnosis and intervention of brain disorders. And the other arm is to develop brain-machine intelligence technology. This is an area of great interest to the information technologists. And we call this structure a one body, 2 wings, a structure that provides a basic framework. And for particular interest of this meeting, I would like to talk mostly on the applied component of early diagnosis and intervention of brain disorders. We feel that there are major disorders that inflicting upon society, in Chinese society and globally, that include developmental disorders and the psychiatric disorders, and neurodegenerative diseases. And to deal with these diseases, early diagnosis and early intervention is generally agreed to be the most likely to be effective. And we think that this would start from the understanding of disease mechanism, the genetic and epigenetic factors, and various pathophysiological dysfunctions of the circuits. But the important aspects to develop early diagnostic tools. And many of this has been currently ongoing around the world, and that include genetic, molecular, brain imaging, and cognitive functional tests that provide markers for the disease. Once these are identified, we would have to find early intervention approaches. Now, pharmaceutical approaches are obvious. The approach comes to mind, but we also should pay attention to the potential physiological and physical modulation that specifically address the dysfunction of a particular circuit. And this may provide a very effective treatment approach in the future. And finally, we need to develop animal disease models using non-human primates, which is going to be very useful for, especially in the development of intervention approaches that may need these animal models. The Chinese effort will be focused on macaque monkeys. In comparison, the Japanese project is now focused on the marmoset. So both countries, or the Asian scientists, will be developing many useful disease models, including those for Alzheimer's and Parkinson's, that can be used in the future by the medical community. And the clinical platform needs to be established that includes the brain imaging database of a uniform standard, and that can be shared by all the people in China as well as in the world, and blood-based biobank for disorder, national blood bank is also needed. We also need a brain bank, which is greatly in demand. That including repositories of both healthy and diseased brains. Finally, we think a very important component is the national center devoted to health training, brain health education that can be widely promoted in the country for promoting early for the prevention of brain disorders in the general public. Finally, a few words on the applied component, the brain-machine interface is one of the technology that needs to be developed. And the electrical and magnetic ultrasonic neuromodulation technologies. Both of these are likely to be used for medical applications. And there's also efforts that are going to be devoted to the development of brain-inspired network models and computing methods. And these are the software for the artificial intelligence. And also brain-inspired computing, processing, and storage devices, the hardware of the artificial intelligence. Finally, the brain-inspired robotics. So that's about it. The one body, two arms, that's the China project. We expect that we will have great impacts on the global brain science and medical community. Thank you.
Okay, unfortunately, we cannot have questions. This is the barrier of present high tech. And we come to the next presentation.
The next presentation is from the Latin American Brain Mapping Network, and I believe Edson Amaro is to be the presenter. Edson, please.
Okay. So thank you. I'm here representing Latin American Brain Mapping Network, and actually The current chair, it's Valeria, but she kind of convinced me to do her job, and Pedro as well. I have to thank both of them to help me to prepare this. And without further ado, let's move ahead. So my idea is to discuss a bit in 5 topics. First of all, what's LabMan, and then go a bit on the translational potential for the region, the project roadmap that we have, and some challenges and what we need to accomplish those. So LabMan actually started from a meeting that happened in Cuba in 2008, and from that meeting we devised a few ideas targeting the progression of methods and also promoting student exchange and overall trying to contribute to build basic and applied science and to help for human brain mapping in Latin America region. So that's a publication that tells a bit about the story. And from that, we also managed to keep the conversations and end up doing a website, which was an effort from many labs in the region. And in 2015, we end up as the first chapter of Organization for Human Brain Mapping. And since then, this year, we had the first election, so now we are having every year another chair for this chapter, and we're also more enthusiastic about the groups that have joined this network, which is basically a network to exchange ideas, as I said, students, and promote human brain mapping in the region. So there are 28 research centers in Latin America. Each one is developing their own research line, and there's lots of integrations between them that are still not happening, and some that constitute the projects that we have been now conducting with funding approved across countries to promote, again, different sorts of research. So we're pretty happy with that. So we're in a position now to move ahead with this initiative for our BRAIN project. So in regard to the translation potential, first of all, I'd like to highlight some points, and these are particular to the region, I believe. First of all, can you guess how it's living in a shantytown like that one, in conditions that are not that favored to human living in general? And if you develop in those conditions, how would be your life? How would be your brain? How would you understand how things are moving around you? And not only that, that will result ultimately in changes that are dramatic. In this picture, we're seeing a 6-year-old normal child on your left side, and on the right side, the same age kid, but living in an extreme neglect condition. And if this is the case that we need some challenges to develop our neural system, too much challenges actually beyond the limit. And in fact, not only that, you have a kind of window of opportunity, and that it's happening very early in life, so we have to intervene very early. In that graph, what we see is that if you intervene very early in those kids with extreme neglect, you can get them back to their original structural brain size at least. And not only that, if you wait too much, then the damage is done, basically. So this is something that we all know. And there's more. We don't need to look at the brain structure. We can look at what they produce, for instance, speech. In this paper, actually, from Siddhartha Ribeiro, from Natal, what they did was to use natural language processing analysis from the output of kids telling stories when they were looking at cartoons. It's a dramatic change in terms of semantic relationship and also complexity. And beyond that, you can imagine that these kids are using their mind in a different manner. And this can be quantified by their speech only. No need for that sophistication, only computer processing power. In a meeting that was held in 2011 in a first Latin American School of Education, Cognitive and Neurosciences, there was a long list of topics discussed, but basically what was the major thing was to move from mechanistic understanding of the brain based on animal models. Again, in this example, we see how genes expression are modulated by the amount of sleep or sleep depression in animals in the lab. But also on the right side, the behavior of kids, relatively large number of kids, around 200 in each group. One was allowed to sleep, take a nap between class lessons. The other one was just walking. This was controlled in the same class, same school. And basically what you see is that there's a huge difference in memory retention. So remembering that we can translate things from labs to habits that we socially convey to the human beings, like kids in school and we at work. And of course, there are many factors influencing the way education modulates the brains and vice versa. But before climbing a tree, it's really important to collect the low-hanging fruit. And we all know that sleep, I just gave an example, exercise and food, some of the features that can be the source for perhaps programs targeting to improve development. And in that particular case, we perhaps don't know what type of fruits are the best for each kid. We know that there's a relationship between their genome and their expression, and we also know that according to their cultural level, that there must be some differences in the way that you could approach them. So from that perspective, I think if you're interested in this paper again from Mariano Sigman from Argentina and Siddhartha from Brazil, they highlighted the point that there is an opportunity, and actually now when we have the computer power that we do and the amount of information that is available worldwide, we can really target those possible low-hanging fruits by intervention programs and at the same time learning from neuroscience about it. In Chile, there's also one program that is targeting the link between neuroscience and education, and this is again one of the initiatives that we see in the region. We will see many examples that I'm going to give you on spot areas in which this interface is developing. In Brazil, there's a large network that was recently assembled trying to guide the health politics in terms of describing what would be the best methods to teach kids in our current days. And one interesting finding was using this grid and network analysis between scientists and targeting basically the link between neuroscience and education. What you find is from around 25,000 supervisors of PhD theses, we have this distribution that most of the people are either on one side or the other side and just a few in between. And actually, this is an available tool on the internet. You can zoom up and search for a specific scientist and see what are the links between us. And this is a nice tool that I'd like to expand for the whole Latin America and for those that are interested in doing this. Well, in Argentina and Chile, there's also another interesting project that's actually using tablets that are given to the kids in school, and at the same time that to teach them using different techniques to, again, target specific goals based on neuroscience knowledge and to improve learning, and at the same time that to teach them, they can feed back you some information about how they are learning. And in this project, basically, it's part of the one laptop to every kid project. This is the picture from Mariano Sigman, and I believe that this is an interesting link. At the same time that you have this real-time interaction between countries and kids, you can also not only learn, but also teach in a better way and in an individualized manner. And this is an example from Cuba in which this hierarchical and stratified model can be used since a kid is born. So basically, in this case, identifying hearing-impaired kids, and this is done at the hospital when they were born, and then at the polyclinic, and then scaling up to when it's needed to have a surgery for a cochlear implant. So you can imagine that all those labs also are producing information that can be used to better understand neurodevelopment. In Barbados, the nutrition study, it's a fantastic example on how you can put together research in healthcare and also neuroscience. In this particular case, which is a slide given by me by Janina last night, thank you. And the idea is to follow these two cohorts of generations. One that was severely deprived from early malnutrition in the first year. They were then suffered intervention and recovered and then had adequate nutrition throughout their lives. And the other cohort that didn't suffer any malnutrition since they were born. And what we see after 5 to 24 years and also from 40 to 45 years, a huge difference in attention deficit problems, conduct problems in adolescence, maladaptive personality traits, depressive symptoms, lower IQ and school achievement, cognitive inflexibility, And you can imagine health problems, of course, obesity, diabetes, etc., and also socioeconomic status, which is lower in the malnutrition population. When they look at the intergenerational effects of malnutrition in DNA methylation, there was also some evidence in epigenetic differences with 102 genes differently methylated. Some of them, majority, was hypermethylated and a few hypermethylated comparing the two cohorts. So this is, again, an interesting model that has longitudinal data set that might help us to expand this sort of study to other populations. In regard to aging, what we see here is one example of one study that was funded by the Organization for Public Studies, and this was in link with Pan American Organization of Health. Basically, they funded study in 7 cities in Latin America, and basically in this study we collected whole genome analysis from a sample that is based on epidemiology criteria from São Paulo City, and also compare those with healthy aging 80-plus subjects. I'll just cut a long story short, but basically what we saw in that particular study, when we measured the white matter lesions using adenine type protocol, all subjects were collecting the same 3T scanner with 32-channel head coil. We saw that there was an increased number of lesions compared to the study from Scotland, the Lothian study, when they followed this cohort since 1936. We see more lesions in our population. We also see more volume of those lesions when we compare to the cohorts from Rotterdam studies. So this might be something that has to do with the population it's seeing. There's also some very interesting effects on genomics, basically because we have a huge variation in Brazilian population and also other countries in Latin America in regard to the European and African ancestry. And this means that we have a large variability in our population, and that can also give interesting results like this one. For instance, in Brazil, African ancestry protects against Alzheimer's disease, which is kind of a contrasense because most people say that people with the Afro-American origin and basically with darker skin, they have more propensity to develop Alzheimer's disease. This was a well-known information literature, but when you look at the African ancestry, it's actually vice versa. I'll skip that in the interest of time. This is another interesting point. In this cohort, the literacy rate can go up to 38%. Remember, these people are old age people, so they had no access to education. And this means that when we look at their brains, we have a different leftward organized lateralization compared to other studies. In regard to the Cuban project, the approach was interesting for the Cuban brain mapping projects. It is a hierarchical approach in which they first start sampling clinical information, then laboratory sampling, then EEG and MRI in a hierarchical fashion. And basically, 1/3 of these subjects, they had hypertension. And the brain was the earliest and most damaged organ. And this was interesting because in 20% of these cases, they had no demonstrable cardiorenal damage. And those cases would have been classified as without hypertensive target organ damage if we didn't look at the brain. So now it's a quick split, the roadmap. So basically, we don't have a whole region brain project mapping, but we are targeting education and aging. Basically, this has been emerging in parallel in some countries, and we hope to integrate and scale up all projects in education and aging, and to provide complementary information to other brain projects. So again, I think we will have to add is to scale up interventions, providing information at the same time that has helping the population. We are an interesting and very important test bed for health policies for targeting equity. Stratification and hierarchical approaches should be one of the major drivers. And of course, we need to integrate database and analytics. And this is just one slide given by Pedro exemplifying what is his point in which we should actually use low cost and wide available screening techniques and then summing up to more sophisticated technique and then targeting the action in a more rational use of resources. Some of the challenges, as you imagine, we have insufficient funds from our home countries, our governments, although neuroscience is very fashionable, our countries and basically our governments are not aware of the impact of neuroscientific research and even less in terms of translational potential. And there are some key problems. I just highlighted the educational problem that we saw and also the population is aging very fast, much faster than Europe aged in the past centuries. And just in case, this is one example from San Diego in California, in which what happens if you have abused when you are young, and there's huge consequences for the society. So this is in the developed country. Can you imagine in underdeveloped countries? Okay, so yeah. What we need is basically to have intelligent resource allocation processes. So we don't think that the same recipe works for all. So we need to look at different population. And we're talking about the largest population in the world is in the developing countries, not in the developed countries. But the major brain maps that you just seen, they are in developed countries. With that, we believe that the pathway to achieve this goal is to use the resources to boost our visibility with many institutions. Research on health equity combined with neuroscience would be interesting. Integration with other brain projects, of course. It makes no sense for us to start things that you red guys doing much better than us, and we hope that we can also integrate this group with some technique that we've been developing. And data mining systems, these are also fundamental. So we are all talking about big data age, and why not just join in and without creating a different system. So thank you very much.
Thank you very much, Edson. Will you be willing to answer a few questions if there are? Stay on for a little while. Any questions? If none, then thank you very much.
So we come to the last presentation. It's by Professor Alan Evans from the Canadian Brain Institute at McGill Centre for Integrative Neuroscience in Montreal, Canada.
Morning, everybody. Oh, it's bright. Thank you, the organizers, for giving me the opportunity to speak about the evolving Canadian Brain Initiative. Basically, in the Canadian context, we've been working on developing a national infrastructure for clinical research for between 10 to 15 years. And we're now approaching the second phase where we're rolling out the clinical applications built on top of that in a more formal sense. So I will describe both of those components as we go. So for a large part of my talk, we'll be talking about the infrastructure that was needed to be put in place for such national and international linkages before we go on to the applications. This is what we're all trying to deal with in its essence. We're trying to capture any individual at the level of multidimensional characterization in behavioral, genetic, gene expression, and brain phenotypes to characterize any individual. We're trying to follow that individual through life, and in particular, when we do cohort studies, we're looking to see if that particular cohort is diverging from the normal trajectory and whether or not it breaks down into subtypes of disorder. And increasingly, we're moving towards precision medicine. We're trying to characterize any individual and make predictions about whether that individual is suddenly beginning to move down the wrong path. In our own context, the hub of the national infrastructure is the Ludma Center for Neuroinformatics and Mental Health. And this essentially brings together three major domains: the epigenetics of Michael Meany, his work on the influence of environment on brain development, and the genetic gene expression. My own lab with imaging and behavior, and Celia Greenwood's statistical genetics environment, all bound together by a high-performance computing environment and a lot of IT infrastructure. I will talk about two particular components within that infrastructure. CBrain is the high-performance computing portal, and Loris is a web-based databasing system, both of which have been used to construct a national infrastructure. The good thing about Canada, I should point out, is that it has a relatively large impact on the national stage, particularly in clinical research. Canada probably has the highest per capita representation here at human brain mapping. But it's small enough in population terms to be organizable. We can get pretty much everybody involved in any particular domain of clinical research, neurodegeneration or neurodevelopmental disorders, in one room. It'd be a big room, but you can do it. The infrastructure that I'm describing is being supported by many different private foundations, by the Canadian Institutes of Health Research, by Brain Canada, and increasingly new initiatives. Particularly, I'm thinking of the so-called Canada First Research Excellence Fund, or CFREF, which is putting about $1 billion into research across Canada, and brain research is a major component in that. And they're very much interested in national infrastructures. So CBrain is providing the high-performance computing infrastructure. It binds together about 200,000 processors across the country. And the purpose of CBrain was to make this infrastructure available to neuroscience researchers. Today, we have about 10 million compute hours made available to brain researchers across the country for free. And the essence of the CBrain platform is to provide distributed data, computing resources, and users via a API portal, the CBrain portal, which essentially mediates the interaction between the average unwashed researcher who doesn't know or want to know much about the IT infrastructure and the IT infrastructure itself at the bottom, which may be data servers, execution controllers, that can manage compute resources which may be terrestrial in terms of supercomputer installations, or it may be cloud services. So basically we have an environment where data is collected at multiple sites, passed into database repositories. We use Loris, other people can use their own databasing environments. Those are then passed onto the CBrain platform for any level of compute-intensive analysis using the kinds of compute resources I've shown below. I could go on at length about all of these various features, but perhaps the most important one is that it's a multi-point data transfer. It's not a centralized facility for storage of data or compute resources. Those can be essentially installed on any of the nodes across the network. CBrain is used across Canada, obviously, but it's also being propagated out globally. And as of February 2016, there were 400-plus user groups in 22 countries around the world who are accessing the CBrain infrastructure for their compute resources. If you want to learn more about the CBrain infrastructure, you can go to this publication in Frontiers. Increasingly, we're concerned with interoperability so that our software tools can be set up on CBrain. They can also be set up on equivalent platforms around the world. One that comes to mind in particular is the VIP platform in Europe. And we're right now engaged with the Human Brain Project to align the CBrain platform with the Human Brain Project infrastructure. And that's ultimately we're working towards a network of platforms for compute infrastructures for large-scale global international projects. Second infrastructure that I wanted to briefly mention is the Loris web databasing environment. Loris was originally developed it launched in 2000, so it's been going around for 16 years, over 100 man-years of development. And it allows the storage of multi-domain data. Originally, primarily imaging and behavior were always the main components of Loris, but in recent years we've added the storage of subsets of genetic information. I stress that we don't want to store all of the raw genetic data, but we do want to store CNVs and SNPs and those data which are relevant to a joint analysis of imaging, behavior, genes, and epigenes. We've added recently epigenetic information to support Michael Meany's work in child development. and tissue samples, which are part of the MNI Biobank project. This is the original project that Loris was developed for. And the point that I always want to make about this is that it's not just a repository for upload and download of data in a secure and properly curated fashion. It's also an environment for managing multi-site data acquisition according to a common protocol, as well as being an interface to analysis pipelines, both our own or those, for instance, SPM or FSL that other people have developed. And that's the interface to the epigenetics and biospecimen data that I mentioned. If you want to learn more about this whole ecosystem, you can go to this paper that came out recently in your image. It's important to point out that this infrastructure is supported by something on the order of 25 to 30 IT professionals at the MNI who in close association with the scientific community, and it's always entertaining to watch the scientists in my lab talking to the geeks in my lab. We force them into the same room, and there's some element of cross-fertilization. So, as I said, this environment is now available for data sharing, where we integrate both imaging and behavioral information and biobank tissue samples, DNA information, and we're just starting to look at iPSCs to incorporate that kind of information into Loris. The big step for us in the next year or so is to cross that line from the private database infrastructure within our specific research projects into the public domain. And we think that most of the issues here are policy-wise rather than technical. The international communities that use Loris right now, some of them are shown here, and I'll just give you one or two examples of that. On the Canadian scene, the Canadian Consortium on Neurodegeneration in Aging is somewhat akin to the ADNI project. It involves 32 centers around the country, 48 PIs, 370-odd researchers across the country, who are collecting data in 4 subtypes of dementia: Alzheimer's disease, vascular dementia, Lewy body dementia, and frontotemporal dementia. And all subjects are subjected to deep phenotyping, imaging, genetics, behavioral characterization. All the data is stored in Loris. It will be processed with the CBrain platform as and when it becomes available. It's still in the early stages of data collection. So on the international scene, there are many projects. The Latin American Brain Mapping Network you've just heard about from Edson. I was very fortunate to participate in the first Latin American Brain Mapping Network meeting, basically because Quebec qualifies as Latin, and it gets me out of Quebec in January. I spent a lot of my time interacting with Pedro and Keith Worsley in the Cuban context. And we'll come back to that in a moment, because the next step in this particular story is the so-called CCC network, or the China-Canada-Cuba network, where we're setting up linkages between our own countries and partners in China, many of whom you can see here. And you've heard from Muming Pu, you've heard from Pedro, spends half of his time in Chengdu. I don't know how he does it, to spend half of his time in Havana and half of his time in Chengdu. They're putting in a plane link directly from Chengdu to Havana, I understand. You think I'm joking. The mayor of Chengdu is proposing to put in a link to Havana. So one of the benefits of a project like the CCC Axis is that we're bringing together 3 countries which have very different demographics, but there are benefits to the socialized medical system that you have in all 3 countries. We can get a lot of data to study specific disorders. As one salient example of this kind of approach, in the West, dementia is predominantly of the Alzheimer's type. In China, it's predominantly of the vascular type, vascular dementia. Well, why is that? Is it genetics? Is it nutrition? Is it lifestyle? putting these cohorts together will allow us to analyze this kind of question in a way that has been hitherto impossible to pull off. Cuba, on the other hand, is I would argue, probably leads the world in the translation of brain mapping research findings into the clinic, into the community. And so putting those three countries together, we think we have very complementary technologies and capabilities from the three countries, which I think are of relevance to the World Health Organization initiative that we're discussing. Back to the Canadian context, we have spent, perhaps we've gone about it backwards, we've spent a lot of time putting in place the infrastructure. We now have a number of programs, national programs, which use this infrastructure. I've already talked about the CCNA, the Neurodegenerative Network. We also have networks, national networks in developmental disorders, autism and related disorders, neonatology, Parkinson's disease. These are all national in scope using the infrastructure that I've described. A major initiative which is presently underway is the so-called Healthy Brains for Healthy Lives initiative with McGill at the hub, but it's a national network that is putting the emphasis on neuroinformatics and modeling, essentially mapping methodologies, at the heart of a $200 million initiative which will focus on the applications of this general schema that I put up in the first slide, the application of that machinery to different types of disorder. At the bottom, we're looking at dynamic brain. The Dynamic Brain Project is looking at plasticity and resilience and how child development can deal with adverse childhood events. Some children are resilient, other ones are vulnerable. Well, why is that? The open science and conventional drug discovery is to use this machinery to target a rational development of drug design and drug discovery. The cultural psychiatry and critical neuroscience component is going to look at the integration of social science metrics with neuroscience metrics. We all know that our children are affected dramatically by Facebook and too much TV. How do we operationalize that? How do we quantify that? How do we take those metrics into a multivariate big data analytic approach to characterize the influence of various social determinants on brain health? And then the last one is looking at the biology and prevention of early developmental disorders, psychiatric disorders. So in summary, the Canadian Brain Initiative is, will have a launch workshop in September, and we hope to have a formal announcement of the Canadian Brain Initiative sometime before the end of the year. It will have a translational focus, which we think is a Canadian strength, to complement major projects that you've heard about today in the US, in Europe, in China, in Japan, and Latin America. And it'll be large enough for a global impact, but small enough to be organizable. This is the mantra for the Canadian approach to the problem. We have a mature national informatics infrastructure. It's important from our perspective to provide bottom-up support, but allow specific subgroups to self-organize and make use of this infrastructure. So we want to potentiate, not dictate how they evolve. We will, of course, provide the analytic infrastructure. And the program is intended to foster open, collaborative projects at different levels and provide cross-disciplinary training and career paths for those people at the interface between information science and neuroscience who often don't fit into any particular category, but they are absolutely central to the evolution of this field. Thank you very much.
Thank you, Alan. I think we have seen in all these different projects a red file. It's neuroinformatics, which is an important part of all these projects. And neuroinformatics can directly address the outreaching to low-income countries and to get information about the worldwide burden, not only the burden focused on the high-income countries. And we have also seen that brain mapping is a major aspect. and the different modalities of brain mapping play a major role in all these different projects. Before we come to the general discussion, are there any questions to Alan? Any specific questions to the Canadian Brain Project? No. Before we had questions which were postponed, and now all the presenters are available here for a general discussion, and I would like to ask you to come forward to the microphones and to make your questions and discussion. Yep. Tanya.
This is all tremendously exciting. And one of the things, on one hand, there's an obvious question here. On one hand, there's the World Health Organization with the word world. And on the other hand, we've heard really wonderful talks from the US, Canadian, Chinese, Japan, and LabMan, different organizations. And I know that, and maybe in the future we'll have a UK brain project, hopefully not. But I know that researchers, and you see that in the talks, that they operate really without borders. There's a lot of collaborations across. But the question is really about, on the funding level, how much interaction is there between the different brain projects, kind of from the outset, to work together for a world brain project?
I perhaps can start the discussion on that one. Just recently we had a global brain research workshop in Baltimore, Johns Hopkins. It was attended by Walter, Katrin, Mu-Ming Pu, myself. I forget if anybody from Latin America was there, Pedro. But it was a start. And there has to be a lot of discussion because for sure there's a lot of reinventing the wheel in multiple countries. But it's rather like watching sausages being made. Sometimes it's pretty ugly to watch, but it tastes pretty good at the end. And there will be a lot of overlap and interaction between these sites as we move forward. This initiative with the World Health Organization goes a little bit beyond that with the focus on translation into low and middle income countries. So the debate is actively underway.
Is there...
Well done.
Can't hear you.
Walter.
I can.
Yes, the NIH has been interested in trying to discuss, trying to build international teams with other funding agencies, and we currently have agreements with Australian Medical Research Council to fund, and they will fund Australian research as part of the brain teams, Lundbeck for Denmark, and Brain Canada Foundation for Canada as well. And we've had discussions with CIHR in Canada and European Union most recently. So we're trying to do that because, as you said, it's an international. Everybody's, it's an international project.
Yeah. Is there, since tremendous amount of data that will be generated from these projects, and there's been a lot on infrastructure, is there talk about harmonizing that with the move towards more open access, that bringing the different data from the different projects together in one large infrastructure? Is that being discussed at all? Or?
Yeah, I think so. I think I don't know if Kat, do you want to mention the data structures that you have? I mean, they seem to be the most advanced at this point in time. But they've been built to bring in data. My understanding, they've been built to bring in data from all different sources. But I don't know, okay. Dr. Ahrens, do you want to talk to that?
Yeah, I mean, in the Human Brain Project, we are building an infrastructure, and this is not an infrastructure which will store the data on one single node. So the idea is rather to have a distributed, a federated system in Europe where you can access the data, for instance, in Jülich, at Cinica, or in Barcelona, so to have the large places named. We are also using cloud systems or the EGI database. So the idea is really to rely in a flexible way on these systems. And we have started, as Alan said, to talk about neuroinformatics structure, making formats compatible, making the different data somehow to be integrated. So I think we have the tools, they are there, but now the artwork is, I would say, really to make it one big endeavor. And this meeting today is for sure one of the steps forward, and we have in the US a meeting at the end of this year where we try to tackle this question. But this is really a huge, number of different questions regarding very technical, so which data format use, where to store the data, how to have access. This is also ethical question, how to make sure that the data are secure, that privacy is an issue, and also how to make the data available. Yeah? So you do not want to wait hours until you can get access to the data. I think in the Human Brain Project, at least, we will develop such a neuroinformatics system where you have a unified portal, which is called Collaboratory, where you can access different types of data which is available in the Human Brain Project. And we will now make sure that it is linked to Canada, to US, to China, and so on and so forth. But this is really a huge endeavor, and I think it's a project which will occupy us for the next years.
Yeah, just to reinforce the point that there are many initiatives going on around the world to explore data formats, integration, interoperability, etc. Everybody's well aware that these are ultimately limiting issues that we have to resolve if we're going to get to the interesting science.
That's one of the purposes of this symposium here, to discuss exactly this aspect. How can we collaborate? How can the different projects collaborate? And what could be the infrastructure? It's just the first step, but it's thought to be brought forward to the organization of human brain mapping and the community here. Yeah, there was a question.
Understanding the human brain at the speed of thought is a complex problem, and to solve complex problems, of course, we need a diversity of experience, input, and knowledge. So it's really encouraging to see today's panel and the diversity across the world of the teams of researchers that are attempting to solve this. My question relates to the first one, having to do with coordination across these efforts. You've addressed coordination of resources or funding, also data, but there's also the aspect of strategy. Some of the projects focus on basic science and tool development, others focus on translation, and others focus on application. And to achieve the translation from the basic discovery to application, we need to have a strategy in terms of how knowledge from one project would inform the design of subsequent projects. So my question is, where are the forums such as this one where that strategy to coordinate these efforts is occurring on the global scale?
Well, I was looking to my partner here, World Health Organization. It's really a global task which you address, and everybody thinks of this task, and it has to be done urgently. And all the brain projects are really open in an absolutely generous way to collaborate with other brain projects. So this is a unique chance, and we need a platform. And this little symposium here can only emphasize exactly what you said. We need a a global strategy to bring the projects together and to bring the projects together for fighting brain diseases at the end. But before we can do that, we have also to include all the scientific questions which were addressed today. But we need a global, worldwide organization. Each of these countries which support brain projects invest an incredible amount of money, which made other parts of science really active, and they say, why so much money into brain research and all these things? I think we are convinced brain diseases are the major diseases and cause a major load on the economy of each country, and therefore it's good to have all these initiatives, but we have to bring it together. So the World Health Organization could eventually play a role in this development.
Thank you very much. Maybe I should respond because my co-chair is looking at me. I think that's a very serious requirement for the field to develop a common and I must emphasize a global strategy for taking this work forward for maximum gains within as short a time as possible. The strategy could be definitely medium term, but there should be some outcomes in the short term too. I don't think World Health Organization is ready to take that on simply because we don't understand the research that well. So on the actual research side, I think there will need to be some other forum which take this forward to think about what all the brain research projects are trying to do and how to avoid in some way overlap and how to exploit the synergies in some way. However, we are very keen to take forward the idea of the public health gains because of these brain projects, and that's where we could take an initiative if there is a need for that and demand for that, to take forward the coordination for the translational part into public health. And that's something that we are very keen to do. World Health Organization is a public health organization and not by itself a research organization. So I'm trying to make the difference. And for the public health part, depending on the collaboration which is offered by the different brain projects, we could certainly think about that, and then we will certainly think about it tomorrow to see how to go forward. I have another point to make, but maybe we should listen to the other questions and we come back to that. So the next one, please.
Just at a very quick point, is one thing I believe WHO can help us. What would be the major questions that would be interesting to be investigated by this community, in a broad level at least? And I'm sure we'll be discussing those points tomorrow, but this would be a major, I think, an important driver for our activities as towards the global human brain mapping.
Thank you very much. I think that's an excellent suggestion. We certainly can think about the major questions that need to be answered from the public health side and pose it to the brain mapping projects to say, to what extent are you already trying to respond to those questions? And if not, how could you work on them and how could you work together on them? So those are things that we can certainly pose. Thank you for suggesting that. Next question, please.
Mine is specific to the Japanese marmoset projects. So it seems like a great system because of the complexity of the brain, yet now the ability to do genetic manipulations. Is there an initiative or funding opportunity for projects looking at brain development associated with that?
Yes, to some extent. Particularly this in situ hybridization is done actually in P0. And so it's about the newborn marmoset. And also we are now analyzing in adults. That's one thing. And also, there's a kind of the project that focusing on some sort of neurodegeneration and also some developmental diseases. But we are particularly at this point focusing on brain mapping. And as Dr. Chairman said, we are particularly focusing on brain mapping and also diseases. So slightly not in center, but we are interested in that. Thank you for your comment.
Thank you.
I'm all for translational imaging, and I think that there's a global need for global database of brain images in patients with orphan diseases and rare diseases. I think that we have the capacity to collect those images, and this would enormously complement and support and hopefully improve the speed of diagnosis and precision of diagnosis of those diseases. So would you support that kind of initiative?
To whom this is addressed? To Organization of Human Brain Mapping?
Well, to all of you, because the problem is that, technically speaking, the capacity to store those images, those patients who have those rare diseases are willing to basically give those images for education, for research, for basically development of more complex description of phenotypes. The problem is that there's currently no one who would be willing to actually have that kind of scope. That's why I had that question to all of you.
Yeah, I think we have a contribution from the Human Brain Project.
From the Human Brain Project, because you precisely identify this problem. So rare diseases are not yet, as far as I know, addressed in these large initiatives. However,
exactly.
I'm aware of many, or at least of several national initiatives, including Germany, where are such studies being performed on rare diseases, like neurodegenerative diseases.
I'm not talking about study of brain disease. I think about the global availability of brain images, because those patients quite often have quite advanced protocols that are being done, but they're just not being shared.
Yeah, right.
This is true. I think that one of the work that we are doing at the Human Brain Project is to develop such an infrastructure where data from such patients can be at least distributed. So that would be the idea, to make it available. But
and
until now,
when that will be? Sometime in the future. See,
I mean, the infrastructure has to be built, and we have until now in the Human Brain Project at least not so much data available. So let us do the first step. Let us use the infrastructure. Let us use the resources that Ellen has described, and then bring in this data. I'm aware that this is a very late answer, but still, I mean,
let me just
have a
give you the framework that those people are operating in. Most of the rare diseases have no cure or treatment, but there are rare diseases that involve the brain. Let's say, for example, X-linked adrenoleukodystrophy. And those patients really would benefit directly from having faster diagnosis, and that has a lot to do with brain imaging. So I think that you can have both very broad, like I think what you are doing is like take all of those diseases, but you can also have very pointed databases that would look into specific diseases.
Yes, but the strategy is really to develop an infrastructure which is capable to host all the data. And you have John Ashburner, for instance, sitting very close to you, who is part of the medical informatics platform. And I mean, this platform is collecting data from the hospitals, including also rare diseases, but we really had to set up the infrastructure at this point. This is a challenging project.
Sure.
Alan would like to say something to it.
Yeah, before you go, so the NIH runs what's called the Rare Disease Consortia. So there's I think it's about 60 rare diseases that are funded, and there actually is a leukodystrophy rare disease consortia. I think Florian Eichler at Mass General runs it, so you could, if you're interested in that, you could get in touch with him.
Mm-hmm. Thank you.
So I think the question that you've asked it has to be addressed at two different levels. first of all, the technology, the methodology, the infrastructure. To a large extent, that's common, regardless of which disorder you're interested in.
Exactly.
Then quite separately from that is something which is specific to rare disorders. You just heard from Walter that there are groups which are looking at specifically rare disorders, and really the burden is on the people who are interested in rare diseases then to form consortia, to write grant applications, to make use of that common infrastructure. I don't think it's really feasible for an organization like OHBM or WHO to take responsibility for rare diseases. You have to do that. We can provide the infrastructure and the common support mechanisms.
Okay, thank you.
I just want to emphasize again the interaction between the brain mapping and brain project community and public health, let's say with WHO at the head. This is very important. There are things each of the side is good at, and it's their job, but the information has to flow. We have to make our results, and we're starting to do that here, or our research objectives known to the health community so they know where we're going, and in fact where they can make suggestions. On the other hand, public health is a domain of its own that has its specificity, and we need knowledge from them. Let me point out one thing. Many projects fail, and I've been to conferences organized by WHO or by the 80-20 conference, which is why biomedical research, 80% of the money only benefits 20% of the population. And one of the major problems is that there's not enough research into health systems. So some of the technologies and things we're thinking about, they depend on how you're going to provide health delivery, which changes from country to country. But it's an important component. I don't think we can reasonably ask anybody. I mean, there's always going to be individuals from the brain mapping community become familiar with this, with the economic aspects, whatever. But one thing that would be beneficial is to provide this information. Now, I do not believe my center, the Cuban Neuroscience Center in Cuba, we go from very basic research in abstract mathematics to actually overseeing health programs. Some of the ones that Edson showed, we've even spread out, let's say, to Ecuador and screened 500,000 kids. Now, we do not believe that that's a link of things that goes from basic research to applied research to development, then to development of programs. We do not believe there's a tight link between this. This would have killed our institution if we said, okay, basic research people, you have to focus on the end result. But what we do think is important is that there should be information flow so that somebody doing basic research can pick up quickly on a result that might benefit the other guy. And this is where I think WHO can help us, the brain mapping community, enormously. I think that, as Alan and others have pointed out, we are here to raise consciousness and to identify issues and to proclaim them and to discuss them again. And then other people have to take this to heart and actually implement it. It's a different job. But I think this first job, my professor in oncology in Cuba, he used to say that if you want something done, you had to talk to it, you had to tell people about it at home, at the bus stop, in the cafeteria, and then after a long while, everybody started talking about it, and then it got done. And I think that's what we're starting here.
Thanks, Pedro, for this view on our discussion today. This symposium reminds me, it's like a déjà vu, on the beginning of human brain mapping. At this time, a few people came together who were interested in human brain mapping. And we never thought of a society of 3,000 people or more at this time. We just started. And step by step over the last 21 years, we got this wonderful society which is capable to pick up even such a question which seems to be, at first glance, outside of our core mission. But thanks to you and Dr. Saxena and the other organizers, we have now this topic on our agenda to discuss what can human brain mapping contribute to those questions without leaving the core field. Yeah, we will not say that we all have now to do research on global health burden and all the problems involved in that. That's far beyond what our society can really give to the community. But what we can give is the awareness that there is more than a strict scientific approach to a single question. We will keep scientific approach. That's our mission. But we will open the perspective. What does it mean for conditions we have in low-income countries? What does it mean for global health burden? And how can we strengthen translational neuroscience from our basis? And I hope that this symposium is the beginning of a permanent discussion within Organization of Human Brain Mapping. I think it's a duty for us to think about this aspect. And our membership is really worldwide, and we have members from many low-income countries. So also these members should be stimulated to bring the topic on the agenda of the Organization of Human Brain Mapping in the future. And I look forward that the meeting tomorrow at the headquarters of the World Health Organization will bring forward our discussion of today. Would you like to see?
Before I make a couple of comments, I would just like to think of inviting all the speakers. If they have had some chance to respond, but if there is any additional points that they would like to make, this is the time.
Yeah, here's somebody.
So,
well, I wanted to commend those who've participated in this for bringing this together, but we're obviously speaking to the converted, and at a time when resources are quite constrained, and but the opportunities have never been better. And the question is, how do those opportunities get leveraged? How do these efforts that are occurring in a multi-modal way, from marmosets to humans, etc., get even more impact? And as Holly said, the strategic opportunities of pulling this together are occurring, in a sense, because of these conversations, but they're not being maximally integrated because there is no homunculus to sort of organize that yet. And I wonder if one of the things that the World Health Organization can do is to provide the bully pulpit to enunciate that not only is the burden gigantic, but that in the World Health Organization's opinion, the traction is now sufficient to make this really a proximal target for a global approach. So the Gates Foundation, it's my understanding, very naive perspective, basically looked at this many years ago and decided, it's not ready for prime time. We're going to do malaria, we're going to do other things where we can get more impact because mosquito nets work or what things like that. The question is, has the progress been sufficient to say to Gates, to say to MacArthur, wait a second, some targeted allocation of resources really could produce an impact? So I would submit that would be a reasonable thing to entertain, because that's part of what needs to get out of this room, this message of the potential and the vibrancy of the work that's going on needs to be communicated beyond our own, small perspectives.
Thank you very much. Any of the other speakers would like to make any comments? If no, then actually this last comment leads very clearly to my conclusion comments. Yes, we need to make the need for brain research much more visible to the world and also to the funders. My co-chair made a comment earlier to say that neuroscience research is being funded at a very large level to the concern of some of the areas. I personally do think, and WHO does agree with me on that there is much more research needed on neuroscience and brain. So there is absolutely no doubt about that. That also leads me to another observation that the funds available for research in neurosciences need to be there. The research itself could be expensive. It could be less expensive. It could be more expensive. But the objective should be to create solutions which are less expensive. And here I make a clear difference between research being expensive and the solutions coming out of that research being expensive. So we are very much on the side of finding out solutions which could be less expensive, which could be simple enough to be used all across the world. And if expensive research is needed to do that, we have no problem at all. So that's exactly the objective of this. Whatever research is needed to produce solutions which will decrease public health burden of mental, neurological, substance use, and developmental disorders, to be applied on a very wide scale, to be applied across countries and communities to all people who may or may not be able to afford that at an individual level, is the need for that. And that's what we are arguing for, and we are happy to raise our voice with you to have more resources along with better coordination so that these public health objectives could be achieved. Another point is creation of knowledge versus the utilization of that knowledge. Obviously, creation of knowledge is important, and one would not have any argument in saying that there should be more funding for basic science research, which makes us understand the brain better, because a lot of desired and in some cases completely unthought for results will accrue from that. And there is no doubt about that. Basic science research is absolutely necessary. It must go on. But a fair percentage of that research should be to the areas which can have early, immediate, and widespread returns. That's exactly what we'll be arguing for, so that the portfolio of brain research could include those elements which are likely to have a predictable, more immediate and more widespread returns in terms of public health gains. We would argue for that, and we are willing to work with you, all the brain mapping projects as well as with the association, to raise our voice with you to do that. We have no money to give you. We don't expect you to give us money, but we can raise our voice to make sure that the funders that there are listen to this. And also my appeal to you that let's keep this as at least one of the objectives in our research. It may not be the only objective, of course. We have talked a lot about the global cooperation. I think that's absolutely necessary, and I see that this meeting itself is attended by a very large number of members from a very large number of countries, which is very good. But even at the organizational level, some amount of strategic thinking about how we can benefit from the experience of research in several countries. Most of these countries obviously are developed countries because they are the only ones who can afford research, can be working together. But also the point raised earlier, how can some of the people living in those countries which cannot afford this kind of basic research can still benefit from that by getting access to databases which they might have the capacity to analyze for those objectives which are very important for them and may not be so important for some other people, is something that we'll certainly go for. So we need to empower the people who don't have still the capacity to do that kind of research, but still being able to see what is happening and to some extent, benefiting from that by getting some side projects out to analyze the data in a way that will benefit them more. And obviously, they might have experience to enrich the basic research in terms of collaboration. So these are some of the things that at least I have learned from these presentations, and I want to thank each one of the speakers, including the people who commented, for sharing your research, sharing your experiences and views with us. Thank you again very much, and I pass it back to my co-chair for any last words.
Okay, the very last words. Thank you very much to everybody. And we have learned that there are some topics which must be better reflected by the different projects, and especially more reflected by the discussions in our council of our society, to see what we can do for the translation of high-tech research to research with less expensive instruments, and finally to give a chance to provide profit for the health problems. I think OHBM is now aware of this problem, is at the very beginning of it, and I hope that we have similar discussions in the future, because precisely this was the way how OHBM developed by itself, from the discussion of a few people about a few problems to this organization, and we would like to support it. Thank you again for coming to this symposium, and to everybody who contributed to the discussion here. Thank you.